our Interim Vice Chancellor and Principal for the University of Pretoria, Professor Mosia, our Secretary General for the African Research University Alliance, Professor Arieti, and the attendees of the ARUA Deputy Vice Chancellors that are here from both locally and from other parts of Africa, our esteemed speaker, Professor Salim Abdool Karim, our distinguished guests, those are here with us in person, and of course we have something like 400 people that registered as online attendees. Good evening, welcome, and thank you to all of you for joining us today. So those of you that do not know me, my name is Sunil Maharaj. I'm the Vice Principal for Research, Postgrad and Innovation here at the University of Pretoria. And of course, it is indeed my pleasure to act as your program director for this, I think, historic occasion this evening. I think it's a singular pleasure for us at University of Pretoria to host this first ARUA Distinguished Public Lecture here at our Future Africa campus. I think when Prof. Arieti asked us many months ago, would you guys be keen to do it at Future Africa? And we said, yes, indeed. I think it'll be an honor for us to do this event. And thank you very much for entrusting us with this particular event. Future Africa is the University of Pretoria's what we call our Pan-African Collaborative Research Platform that works across different scientific disciplines, and we use science in the broader sense, including social sciences, economic sciences, and so forth, with society to address Africa's what we call biggest and most urgent contemporary challenges. Its fundamental purpose is to develop and unlock the potential of African sciences towards local and global transformation. So I think you'll agree with me that looking at the facilities we have here and of course the purpose we try to achieve in this particular environment, there could have been no more appropriate venue for this inaugural ARUA public lecture, where we will be addressed by a speaker of exceptional stature and well recognized globally. Firstly, however, it is my pleasure to introduce our Interim Vice Chancellor and Principal, Professor Mosia, who will deliver the welcoming address on behalf of University of Pretoria. Professor Mosia.
Thank you, Professor Maharaj, and good afternoon, everyone. Good afternoon, how? Yeah, we are happy people. We are happy people here. You know? We're happy people. We're not serious, you know, that seriousness. We'll only be serious when we hear the keynote address, because that's quite profound. It also gives me a great pleasure indeed to welcome you to this great university. And this is a very, you know, momentous occasion for us to host this inaugural ARUA Distinguished Lecture. You know, we are, of course, expecting quite a lot of enlightenment and very instructive comments. And I know that we'll leave this auditorium, or even those that are online, you know, pretty excited and positive about the lecture and what the future holds for us. Now, ARUA, as we know it, you know, it's a collaborative platform, really, for institutions such as ourselves and others. And it is where we actually have the exchange of these great minds to actually advance the agenda, you know, that should bring a positive difference into humanity through science and innovation. So we know that, among other things, our focuses on graduate education or training, you know, because we need to build this cohort. As you can see, we are getting old. So the more you get younger people and younger scientists, the better for us. And this evening we're honored, indeed, you know, to also host, you know, one of our very own experts, on infectious diseases and epidemiology and of course global health research. You can just see he's indeed quite a luminary. Professor Salim, Abdool, Karim. You know, when we had COVID and then we were in captivity, you know, and and the only, you know, positive thing and hope that we're getting was the manner in which you were presenting, you know, the data and the information. And you know this, it must have been said 1,000 times to you how you brought hope to us. Even politicians, you know, because, you know, they always are controversial, but many of them were quite positive of what they were hearing. I don't know whether because they were afraid of COVID, whatever. But Prof is quite a trailblazer, and I'm sure Professor Areti will introduce him formally. But, and I've just been wondering, Professor, if I must say, you know, Salim and others, and Sunil, why don't we have, you know, Professor Salim at UP, you know? Because, you know, I know the home is in Durban and everything, and, you know, KZN is a very important university. But all I'm saying is, KZN, I was with a colleague there, just watch the space. Yeah. We are very aggressive when it comes to certain things. But, I mean, you know, he has shaped the world, really, our international and the global guidelines on TB and HIV and a lot of groundbreaking research. I will not go into the detail because you know that this is a man of stature. And we'll listen to him shortly. But as I said, he instilled a lot of hope in us. And we also, the fact that you're also attached to a university makes us you know, a very special community of intellectuals to interact with you. I mean, as we welcome you to this great university, that we know, you know, in one way or the other, or in many ways rather, through your academic work, through your expertise, you have been involved here for many years, and our colleagues have worked with you in different ways, and we appreciate that, and we hope that as we shape the future, because we are really troubled, you know, as a country, but also as a world, and especially if you look at health, you know, and all of these pandemics and things and so forth. And of course, you'll speak about the two pandemics and with your expertise, and we are looking forward to this insightful lecture. And as Prof. Maharaj said, you know, this is our platform where we want to, you know, get as many, you know, intellectual minds as we can over time in different disciplines, because we are a multidisciplinary focus. You know, academics speak of transdisciplinary, multidisciplinary, interdisciplinary, a whole range of confusing things, but we do that. It's fine, it suits us fine, you know. As long as we're able to generate knowledge, such as the ones that you have produced to make this transformation of the continent our lived experience in our own time. And without any much waste of time, because, you know, I had some colleagues who were saying, well, Prof is late, you know, delayed, and the flight, you know, and coming to Pretoria from OR Tambo, it's another journey, you know. It can take you longer than the flight from Durban to here, you know. So, but our attitude is, you know what, what we are doing here is far more important than a minor delay. We don't even mind people to tell us that we are using African time. We are Africans after all, so what? Whose time should we use? And so, Professor, I'm sure that your contribution here, and to all of you colleagues, as we interact, please feel free to engage with these issues. They are very important. They're very serious. And for ARUA to have selected you among other experts to particularly speak on this topic in this first inaugural lecture, and of course we're hosting it, but the most important thing is that we know that your input will make ARUA invite you again in the future, to actually engage us on a number of, because this is not the only thing that you do. We know you do many other things. So, ladies and gentlemen, thank you very much for your time. We look forward. Just relax, Don't tell me now you want to leave. Just hang in there, Even those that are online, This is very special, He would have probably been in Geneva now, but he's here with us. Thank you very much.
Thank you, Prof. Musia. So again, for the online viewers, during the presentation, just to remind you that you'll be given the chance to post any questions you have on the chat, and then we will do that after the presentation. So for now, it's my honor to ask our Secretary General of ARUA, the CEO of ARUA, Professor Ernest Arieti, to do a few welcomes and introduce the speaker.
Thank you very much, Sunil. Distinguished ladies and gentlemen, let me stand on the protocols already established by Sunil and the Vice Chancellor to say a few words and introduce our speaker for this evening. In terms of, I'll introduce him by explaining to you why he is the speaker. ARUA decided a year ago to have these annual public lectures. And the main idea was to create a platform that allowed us to showcase to the rest of the world accomplished African researchers. The world believes that all good researchers come from Europe or North America, but we do have in Africa a very good number of accomplished world-class researchers. So this will be the beginning of that process of making the world know and accept that we do have the men and women who can change our world. So the board of ARUA, that is the vice chancellors of our 16 universities, now 17, accepted the proposal to have these lectures. And I was tasked to find a speaker who will set the tone for the rest of the lectures that we'll be having. It wasn't too difficult to decide who that speaker should be, because I met Salim for the first time a year earlier when he spoke at the ARUA conference here in Pretoria. He was then in Tokyo and spoke to us in a hybrid form, and that went really well. I was extremely impressed by the presentation he made, and I thought, well, if this man is as good as he sounds, he should be the person to invite for the first one. And I mentioned it to a number of my South African colleagues, and each one agreed that Salim would be the most appropriate person to begin this. So, while I was debating how to reach him, I was invited by the International Science Council to the World Science Forum in Cape Town. And as I walked into the room, the first person I met was Salim. And I said, can I have a word with you during the break? And he said, why not? You know, I was expecting him to be a typical pompous, the well-known, world-class scientist who would be asking me questions about what I wanted to say, but he never did. And I said to him, we would like to do this. He said, why not? So here we are. I've been extremely lucky in finding Salim to do this for us. He comes extremely well prepared. He's very well known. Everybody who has seen the flyers we've used has said only positive things about him. I can read his profile for you, but I'm not sure if there's a need for it. I mean, but the important thing is he is highly regarded throughout the world. In having him deliver the first one, I have no difficulty whatsoever in using his name for other subsequent invitations. So we can go to the next person for next year and say, you know what, Salim, Karim did the last one. Well, they'll say, yes, okay, if Salim did it for you, then I better do it for you. So that's the purpose, and I am happy, very pleased. Before I take my seat, I'd like to say that the chair of the ARUA board has just joined us. Professor Barnabas Nawangwe is the vice chancellor of Makerere University. He was in Johannesburg for a meeting, but he couldn't afford to miss this evening's event. So traveled from Johannesburg to come and join us. Barnabas, yes, sir. That's Barnabas Nawangwe. He's the chair of the ARUA board. So Salim, let me now invite you to come and speak to us and addressing the topic AIDS and COVID-19, a tale of two pandemics. Thank you very much.
Professor Mosia, Professor Maharaj, Professor Arate, Professor. I see in our midst many friends. I see Dr. Hackman, Dr. Burton. It's a great pleasure and an honor for me to be here with you today. I was pondering, what is it I could share with you that would really meet the fundamental concept of ARUA, which is to be a brains trust. That no matter that we as individual institutions may compete with each other, it's our collective collegiality that will enable us to rise and compete with the rest of the world. So I was deeply honored when Ernest asked me to do this lecture, and I thought that a lecture of this stature needed to bring together as much of my research as I could. So, ladies and gentlemen, I am going to walk you through, in 45 minutes, a journey where I will talk about just over 500 journal articles of research condensed into about 30 minutes. And I'm going to share with you a 30-year journey in trying to understand the HIV pandemic and to find ways in which we can deal with what is the most important question and the most important problem in the HIV pandemic, and that is the high rates of HIV in young women. But I thought, who wants to hear about HIV? They all want to hear about COVID. Well, nobody wants to hear about COVID anymore. But let me just share with you how, almost in fast forward, a research program of 3 years of research that I have been involved in relation to COVID. And I'll end off with just one key lesson that we can take away from both pandemics. So that's what you have in store for you. So let's plunge directly into the pandemic of HIV. So the AIDS pandemic, if we look at the situation globally, we have roughly about 39 million people living with HIV. We had about 630,000 deaths last year. And when you think about 630,000 deaths, that's a lot. Right? That is competing with the number of COVID-19 deaths in this year. Just to give you some idea of the relationship in terms of the numbers. And about 1.3 million new infections. So we are far away from the end of AIDS, which is our SDG goal for 2030, which is the ending AIDS as a public health threat. We are far away from that. And what's important is that Africa bears 70% of the global burden of HIV. 7 out of every 10 people who have HIV in the world live in Africa. And sub-Saharan Africa has about half of all the people living with HIV. And roughly about half of all the new infections that are occurring. And within sub-Saharan Africa, young women, adolescent girls and young women, teenage girls included, have the highest rates of new HIV infection. They comprise about 63% of HIV. So, let me share with you 4 aspects of 30 years of research, because I mean, I just can't do it all, but I chose 4 things, 4 of my favorite topics that we have explored in the course of this pandemic. So let me start off with talking about HIV in young women and the fundamental driving force of the AIDS pandemic and the way in which AIDS is spreading in Africa. So it started back in 1990. That's about 30 odd years ago, when my wife and I came back from Columbia University. We had gone to Columbia University to study, and we came back at the end of 1988, and we both just completed our master's degrees in epidemiology, and we decided we are going to tackle HIV. And one of the first studies we did was a community-wide survey of HIV. There were almost none at the time. So one of the first ever HIV community-based surveys, we undertook it in 1989. And in 1990, we produced these results. These results from one study has shaped 33 years of research on HIV. Because this survey illustrated for us something that we had not known. When you think about HIV in 1990, what was HIV in 1990? It was largely homosexual men. It was all about the challenges in the US and Europe and ACT UP, and that's what it was about. Africa was not even on the radar screen. And we produced this result. And what does this show you? I'm going to walk you through this because it is central to all of the things I'm going to talk about. So I want you to look only at the blue line in this diagram. And this is a diagram of prevalence on the y-axis and age on the x-axis. So if you look at the blue line, in the youngest of the children, 10 to 14, there's very little HIV. There were just a few cases from mother-to-child transmission, but there's really very little HIV. And when you look at the 15 to 19-year-olds, and remember the blue line is boys and men, almost no HIV. But when you get to 20 to 24, you start seeing the cases rise, and by the time you get to 25 to 29, you see a really high rate of HIV. But look at the situation in girls. In girls, from a situation where you have just a very low prevalence in the 10 to 14-year-olds, when you get to the teenage girls, they have this incredible burden of infection. It just rises. The rate at which new infections are occurring drives that line to rise so rapidly. And what this shows you is that in young girls, the bulk of them are going to get infected when they are still teenagers. And that prevalence rate then starts coming down. So you don't see it going up again. So that those who are going to get infected when they're still young. And why is that important? It's important because young girls are the ones that are going to have the children. Young ones, they are the ones in school who are now going to go and study, nursing and medicine and so on. It is fundamentally destroying the fabric of our society when you see this kind of situation with young girls. So we described this in South Africa. Was this unique to South Africa? What about the rest of Southern Africa? Well, it took several years later, more than a decade later, when UNAIDS published this diagram. And in this diagram, they show country after country. The red bars are prevalence of HIV in young women, adolescent girls and young women. And the blue bars are the prevalence of HIV in adolescent boys and young boys. And what you can see, country after country, the red bar is much higher than the blue bar. So this fundamental discovery that we made, which is so elementary, in 1990, turns out to be the vast driving force of the pandemic in Southern Africa. Now, it doesn't take a University of Pretoria professor or a University of Pretoria lecturer or a postdoc student to realize that the young girls are not getting infected from young boys. That should become apparent immediately because the 15 to 19-year-old boys have very little HIV. Which means that the young girls are getting infected from men who are several years older. And this discovery of the age disparate sex that's driving these high rates in Africa was fundamental to understanding its epidemiology. But there were others that challenged us when we described and looked at this issue of age disparate sex. They did other analyses and suggested it's not true. But our evidence was compelling. And here, I'm sharing with you data that my wife, from a study my wife did. Actually, I should make a disclaimer. Almost all of the research I'm going to present is principally that done by my wife. Just so that everybody is clear. So she does all the hard work, and it's all her data. I just stand here and claim the credit. That's my half of the contribution. So she did this study looking at grade 9 and 10 students from several schools in rural KwaZulu-Natal. And if you look at the girls and you look at the boys, you see this picture that tells the entire story. So if you look at the prevalence rates here of the boys, from a low of 1% in the 15-year-olds to a high of 1.8% in the boys who are over 20 years old. Look at the situation now in girls. Drastically different. Already by age 15, 2.6% of the girls are positive. 1 in, 25, roughly. And when you get to, no, it's 1 in 40. When you get to age 20, it's 1 in 4 girls are already infected with HIV. That told us the huge challenge that we are facing. We did another survey, a massive survey of close on to 10,000 people in a rural community in KwaZulu-Natal to begin to grapple with exactly where the main incidents of new infections are occurring. And if you look at the red bars, the prevalence in women, you can see how it rises so much faster in young girls than it does in young boys. And that by the time you get to 30-year-old women, it's about 70% have HIV. I mean, just the devastation that means at a community level. So one of the things we had to do to settle this debate by people who are challenging us that no, we are wrong in age disparate sex was eventually in 2016, Professor D'Oliveira, whose research unit and office is a floor below mine at the Nelson Mandela Medical School, I called him up and said, we're going to do a study where we need to look at how can we draw linkages to understand transmission. And he said, oh, we can do that with phylogenetics. And so we did that. We sequenced thousands of HIV in a community. And looking at the virus, we can say which people have the same virus or very similar virus. So in other words, they're very likely to have infected each other. And we create those as clusters. And then we look at each cluster and we try to figure out, based on the force of infection, who infected who. And what we showed what you see in this diagram, that young girls, especially teenage girls, have the highest rates of infection. They are largely being infected by men about 8.7 years older. It's about 9 years older. These men who are in their early 30s are getting infected from women in their 30s. Almost no difference in the age, about 1 year difference. So what we described is the cycle of HIV transmission. The cycle of HIV transmission, high rates of HIV in young girls. They grow up. When they reach age 30, they're now trying to find their partners, who they're going to marry. They are now partnering and having sex with men in their 30s. Those men don't have HIV to any significant level. They are these 30-year-old women are infecting the 30-year-old men. And those 30-year-old men, 39% of them are having a liaison with a teenage girl. And those men in their 30s are in acute infection. They have just become infected, so they don't even know they are infected. And they have high viral loads, and so they are very efficient transmitters. And it's those men who are sleeping with the young girls that are now infecting the next generation of young teenage girls who will grow up and when they reach 30, will infect the next group of men, who will infect the next group of teenage girls, who will grow up, and you get to see the cycle. That is the cycle that we described. When we described it and published it in the Lancet HIV, within a matter of a few months, UNAIDS produced this report called Get on the Fast Track, and it talks about the life cycle approach to HIV. And what they did was they basically took our research and our nice diagram, and can you see what they did to it? They simplified it. So what they did, they gave it nice orange and blue colors, and they showed you how from young girls it goes to the older women, and how they then transmit to men, and how the cycle continues. When I first saw this, I said, why didn't they just use our diagram? And I realized actually it's easier to understand their diagram. So this became the policy and the priority of many countries in Africa, including our own. This is the cover of the South African HIV strategic plan. The number one priority in the South African plan is breaking the cycle of HIV transmission. And what do you think they did? They took the UNAIDS diagram. Can you see how they changed the color? And they called it infection pathway, and they took ownership of it. This is now their diagram. I've been wondering whether we should patent or copyright our diagrams, so that people don't go around changing them all the time. But for me, it highlighted how, from a basic set of research, we get to a point where we can understand the fundamental driving force of this pandemic and how it can be adopted and become policy in several countries across Africa. Now that we know that young women have these high rates of HIV, we need to figure out what are we going to do about it. It's not enough just to know it. We have to do something about it. And so we did. And it all started in 1990, again, since we produced those results. And we sat down and we said, we got to find some technology that women can use to protect themselves. And the problem was very simple. We had the ABCs: abstinence, be faithful, condoms, and circumcision followed a bit later. But if you take the ABCs, they don't apply to women. And they certainly don't apply to young women. Abstinence, well, that's too late by the time we're talking about. So abstinence is out the window. Being faithful, a young girl can be faithful to the very man who's going to give her HIV. And condoms are under the control of men. And circumcision is a male intervention. So when you are a young woman, as I remember this very clearly, in 1991, sitting in my clinic, and an 18-year-old came to me, and she was telling me how she's worried about HIV. And she's asking me, what should she do? And I have to say to her, actually, there's almost nothing I can tell you except, try to get your partner to use a condom. Because we didn't have anything for young women. And so we set about solving that problem. So I remember we showed that challenge of young women and the age-disparate sex in 1990. In 1993, Kuresha and I set about doing our first study of a vaginal film. We worked with a company in the US to use a product called Nanoxynol 9, which was a spermicide, and we made a film, and we studied this film to see if it prevented HIV. And it didn't. It didn't work really in a group of sex workers we studied, and so we killed that idea. We then joined the World Health Organization in a huge study also of naloxone 9, but now in a gel formulation. A disaster. It doesn't work. So we went on, and we went on, and we went on. We studied product after product. We were involved in making products. We were working with partners in industry who were making products. We studied Pro 2000 gel. We studied buffer gel. We were, by 2010, we had reached a stage where we had tested over 8 different products, and every one of them had failed. It got to a point at that stage that Croatia and I were widely acknowledged as the experts in failure. No one we know had failed as much as we had. Over 18 years of research. Now you would say, that's career suicide. To spend 18 years studying one problem and repeated 18 years of failure, how on earth do you stay and carry on? Because you're destroying your career. Well, the problem was too big. The challenge too great. We had to persevere. And so we did. And so we came across a new drug called tenofovir. Just by chance, we managed to get some tenofovir, actually, interestingly, from Uganda. Our colleagues in Uganda had tenofovir, and we were doing a study on how to treat patients with TB and HIV. And so we got this drug for that study. And when we used it, we realized what a terrific drug it was. And so I flew to San Francisco to go and see this drug company called Gilead Sciences, small biotech, literally one little building in Foster City. Today, it is among the 5 largest drug companies in the world, based largely on the same drug, tenofovir. And I asked them to give me 25 kilograms of tenofovir to make a gel. And so we did. We made tenofovir gel. We chose to study it in a coitally driven manner, and we chose it because of its powerful safety profile and highly effective way in which it suppressed the virus. When we designed the study and we were ready to start, a group of 27 scientists, led by a major figure from the UK, wrote a letter to Nature, attacking us, telling us that this is bad science, taking an antiretroviral drug and studying it. Nature didn't publish their letter, but Nature decided to write an article about it. And they had one of their journalists write it. The title of the article was HIV Trial Doomed by Design, say critics. So Nature and our colleagues in the US and the UK, just a handful, doomed us. They said, what you're doing is never going to work. But there were many that supported us. And we proceeded to doing the trial. And in 2010, we announced the results at the International AIDS Conference. And we provided the first ever evidence that antiretroviral drugs can prevent sexual transmission. What is, you might be aware of, known today as PrEP, pre-exposure prophylaxis, which is with tenofovir. They use a drug called Truvada, which is tenofovir with 3-TC. So we provided the evidence that serves as the fundamental basis for PrEP. The findings were lauded across the world. We were ranked by Science, the journal Science, as one of the top 10 breakthroughs of 2010. We were the Lancet's paper of the year of 2010 with another paper on influenza. Editorials in New York Times, and we were front page headlines in newspapers across the world. But that result that we published in Science and presented in 2010, showing that tenofovir gel prevents HIV infection in women, then led to a whole lot of other studies and research, and there were many other parallel studies underway of tenofovir as a tablet form that showed it was as good, if not better, than the gel. And eventually, the WHO provided a global guideline for PrEP in 2015. So when you think about it, we had the idea, well, we had the problem, we identified the problem in 1990. We had failure upon failure. In 2003, 13 years later, we have the idea about tenofovir. It takes us ages to go and make the gel, do the study. We start the trial in 2006. We provide results in 2010. In 2015, the WHO provides it as a global guideline, and South Africa introduces PrEP in 2017. That's the 27-year journey that it took us to get from the first description of the problem to the first attempt at solving it. But we wanted to understand that it wasn't simply behavior that was leading to young women having such high rates of HIV. It's not simply behavior. It's biology and behavior. So we set out to study the behavior, and so I'm not going to cover that. And we set out to understand the biology. And let me deal with that now. So we showed in this paper in The Lancet that in order for tenofovir to protect, you need a very high concentration of tenofovir. Now we wondered why that was the case. Why would you need so much of tenofovir in order to protect? Well, this study gave us the answer. I'm going to walk you through this result because it's so fundamental. What you're looking at is called a heat map. Each column is one woman. And if you look at the blue square and the yellow square at the top, that tells you whether the woman remained HIV negative throughout follow-up or if the woman got HIV. So if it's a blue square, the woman got HIV. If it's a yellow square, the woman didn't get HIV. Each row is a marker. It's a little protein cytokine in the vagina that we measure using Luminex, and we show where it's raised and where it's not raised. So where it's raised, it's red. Where it's not raised is green. Now, when you look at this heat map, what becomes obvious? Where it's red, most of the squares are blue. Where it's green, most of the squares are yellow. So this picture tells you what would take me ages to explain, which is that genital inflammation. There is a subclinical genital inflammation in young women that places them at risk of getting HIV. Why? We wondered. What has genital inflammation got to do with any of this? And so we did this study which showed that HIV to infect a human has to enter a CD4 cell. It can only infect CD4 positive cells. Well, it turns out that genital inflammation brings the CD4 positive cells to the genital lumen. It attracts the CD4 cells because those are the cells that are coming in to fight the inflammation. So now we understood, oh my goodness, inflammation, bringing CD4 cells. It turns out it also activates CD4 cells, which leads them to express CCR5. They get an overexpression of alleles of CCR5, which is exactly what the virus is looking for. So here we have a situation where we now understand genital inflammation is linked to the CD4s, which are becoming a nidus of infection. So why is there genital inflammation? What's causing it? Well, I don't think we know what's causing it yet. But one of the things we described in this paper in Nature is how human papillomavirus and clearance of human papillomavirus and new infections of HPV increase genital inflammation. So now we have some idea that HPV may be a contributor, but it doesn't explain genital inflammation. It's a small contributor. So we don't really fully understand. But we know that genital inflammation, in this paper in Nature Medicine, we showed that not only does it create the biological basis for infection, it's also impacting the effectiveness of tenofovir. Now we needed to understand that. And that became clear in this paper in Science. I'm going to show you what this paper tells us. It tells us that women who have lactobacilli in their vagina that is dominant. Lactobacilli are bacteria that make lactic acid, and they are the good bugs to have. Remember, in a woman's vagina, there are thousands of different bacteria. In fact, when we do sequencing of the bacteria, there's at least 5,000 to 10,000 different bacteria. But when there's lactobacilli dominance, the vagina is generally healthy and is resistant to other infections. But when the lactobacilli are depleted and there are other bacteria that take over, most of them are bad. And they then create the opportunity for the virus to cause infection. So what we showed in a subgroup analysis, women with lactobacilli have effective protection from tenofovir gel. Women who don't have lactobacilli dominance, they are the ones where tenofovir gel has almost no effect. Now, we were struck. Why? Well, it turns out in this diagram that you look at above me here, one particular bacterium called Gardnerella vaginalis. When Gardnerella vaginalis is dominant in the vagina, it eats up tenofovir. Literally, when you insert tenofovir into that vagina in the midst of Gardnerella, within 4 hours, about half of the tenofovir has disappeared. It metabolizes tenofovir. So even our best efforts to try and provide PrEP, and the microbiome is defeating us. We have to sort this problem out. We don't yet have a solution. But what we do have is that the Gates Foundation, based on our results, created a whole new program called the VMRC, which is about the vaginal microbiome. And what we're doing with them is that we are taking women who have a resilient form of lactobacilli. Those lactobacilli stay. They don't get lost. They don't get replaced. We've taken several strains of those. We've selected a handful of those that really are best. And we've taken strains from the US, from South Africa. We've put them all together, and we've made a little tablet. It's a little capsule with the bacteria in it. And that capsule, we are now just starting the first phase 1 study. That capsule melts in the vagina and releases these good bacteria. Our hope is that these good bacteria will take over the vagina. It's only a hope at this stage. But we have to find some way to fix this problem. And you can't treat it with antimicrobials, because if you treat it with flagyl, for example, you'll kill the lactobacilli and you kill all the bad bugs, but 2 weeks later, you're back to square one. So it's not a long-term solution. So we have to find a solution. So all I can say is cross fingers that we get a positive answer that we can change the microbiome and reduce risk of HIV. And let me deal with the last point I want to make, which is that what we've learned is that when you're asking young women to take a tablet or a gel every day for a disease they don't have, it's a real challenge. They just don't take it after a while. They started, several of them, but they just stopped. It's just too much headache to go to the clinic and to get the tablets and so on. And what we showed is that in the clinical trials, that you get varying results. And the varying results are driven largely by adherence. So if you reanalyze the clinical trials based purely on the good adherers, and there you find that actually you get a very consistent protective result. So adherence is our big problem. So we got tenofovir, we just can't get people to adhere to them. So we have to find a new solution. And here comes a possible solution. So in 2012, we discovered that a woman in one of our clinical studies, her number is CAP256, that's her code. And I'm telling you it very deliberately because watch for CAP256. We discovered that this woman had an antibody that is highly effective and potent in killing HIV. Now, when anybody gets infected with HIV, naturally they will generate antibodies. That's just, when you get infected with a virus, you develop antibodies. But when a person develops antibodies against HIV, those are strain-specific antibodies. They only kill the HIV that infected the person. They won't kill any other HIV. And HIV has a lot of diversity. It changes a lot. And so we have been looking for what is called a broadly neutralizing antibody, an antibody that can kill lots of HIV. And so we took her blood and we subjected it to 400 viruses from throughout the world. And we wanted to know, can her antibodies kill all these different HIV? Well, her antibody kills about 4 out of every 5 HIV in that panel. So now we have a broadly neutralizing antibody. We also gathered that it's a highly potent form of it. So we had to proceed next. We had to make this antibody. So what did we do? We sent a doctoral student to the NIH, to the Vaccine Research Center, and we gave her a vial of blood, and we said to her, see if you can find the B cell that makes this antibody. Now you remember B cells, right? Remember high school biology, you have red blood cells, white blood cells, platelets. In the white blood cells, you have T cells and B cells. Well, the B cells make the antibodies. And each antibody is made by only one B cell. So that B cell won't make any other antibody. So we are now looking for a very rare B cell in this patient. Roughly 1 in 10,000 B cells will make this antibody. So we knew it was going to be difficult. She won't really be able to find it. But we thought it was a good idea. We gave her funding for 3 months, and just before she could leave, we got the call. I found the cell. 1 cell. 1 B cell that makes this antibody. Precious cell. So we take the cell, we clone it in our labs, and we set it up in tissue culture. We then proceed to sequencing the gene that makes that particular antibody, and we realize how good it is, but it's still only really, let's say, Mercedes-Benz quality. we want Rolls-Royce. So we manipulated the genes, and we added some additional genes, we changed a few genes, we made it our better antibody. We then, in culture, made the first 3 grams. Took us 7 months to make 3 grams. You know what 3 grams looks like? You won't even see it if it was on your hand. Took us 7 months to make 3 grams in the lab. We send those 3 grams to our friend Dan Baruch at Harvard University. And we asked Dan, can you put that antibody into your monkeys and tell us if it prevents HIV? And so that's what he did. He injected monkeys with 3 different concentrations of the CAP256 antibody. The 21 monkeys in the control group, because it's a blinded study, all got infected. You can see that in the placebo control. See, all the monkeys get infected. All the monkeys that receive CAP256 are then challenged with this monkey form of HIV that infected all of the placebo monkeys. We have never been able to infect a monkey that's received CAP256. So, we now have some good animal data. Croatia and I take these diagrams and we go to see our friend, Dr. Anthony Fauci. And we say, we need some help. We need to make enough of the antibody to put into human studies. And so we partnered with the NIH, and they made us the first batches of the antibody for human studies. The phase 1 studies are now complete. The products are safe. And we've shown the combination of CAP256 plus VRC07, one of their antibodies. The combination is the best combination we can go with. It has the widest range of viruses it kills, and it's the most potent. And we showed in this paper in Lancet, HIV, that the antibody, when you inject it into young women, produces therapeutic levels well beyond 6 months. In fact, even at 9 months, we still have high levels of antibody. So what are we doing? We are now making a 6-monthly injectable of 2 antibodies to protect women so that they don't get HIV. So now they don't have to worry about taking a daily tablet. All they would need to do is to get this injection every 6 months. We could go to schools every 6 months and offer it to them in a mobile clinic, provide it to young women to protect themselves. So we showed that the antibodies provide you with protective levels, but does it actually prevent HIV? That study is underway. We have so far enrolled 720 women in that study. It will enroll just about 1,000 women in the study to look at whether it works or not. So does it work? Watch this space. Professor Arate will have to invite me again to share those results on CAP256. But it's a long road. And I want to make this point to all of you, because I know that you can get disheartened. I know that you will see failure and you will want to choose something else. We enrolled CAP256 in 2004. She became infected in 2005, actually in early of 2006. We put her on treatment in 2008. We identified her antibodies in 2011. We isolated it 2014. We manufactured it. We did monkey studies. All of this, 20 years later, in 2024, we'll have the results. That's how long this road is. So for those who think that you make major discoveries overnight, I don't know which world those people live in. The world I live in, it's a long, long road because we are making completely new medications. And you have to be ready to take on this journey. Just out of interest, I thought I would share this with you, that Penny Moore and her colleagues at the NICD, who are part of our team, have figured out a way that if CAP 256 actually works in preventing HIV, we can back engineer the actual viruses that led CAP 256 to make this antibody. And we can set up a vaccine. So instead of having to give the antibody, we can actually make an HIV vaccine. It won't be a simple vaccine because it will be several vaccines that have to be given in a sequence. But that's the idea. If CAP 256 works, it creates the opportunity. Now we know what immunity prevents HIV. And then we can proceed to making a vaccine to copy it. So, a long road lies yet ahead. And that road, I'm telling you, will not be undertaken by any of us. It is for the next generation who will do that. So let me end off with Pandemic 2, which is just a 3-year journey. So it's a much shorter journey and quicker journey. And I'm going to tell you about the thing we focused on, which was about the variants. So one of the first papers we published, and this was back in April, May of 2020, we published this in the New England Journal of Medicine, just basically describing one approach to dealing with COVID, the 8-stage response. And for those of you who might have watched me in Easter Monday in 2020, when I presented this on television, I shared how we were planning on dealing with it. In fact, several countries copied some of this or parts of it in their own responses. And a few months later, in June, July of 2020, we published this paper in Science, which was capturing how the COVID pandemic was impacting the TB and HIV pandemics, and just bringing the world's attention to this problem that we were dealing with. But by the end of 2020, South Africa had been the first to discover variants. And it was Professor de Oliveira who actually discovered them. And I remember the day he discovered them, because as soon as he saw the sequence, he comes up to my office. And he comes standing at my door, and I don't know if you know him, but he's a very ebullient character, very excited. And that day when he came, it was late November 2020, quiet, looking dejected, puts his computer on, and he says, "You need to see this." I said, "What?" "This virus is very stable. We've been following it month after month. No problem." He said, "You need to see this." And at that point, we discovered a completely new form of SARS-CoV-2 with 3 major mutations in positions 417, 484, and 501. That changed the world. And variants, as we learned, had changed our understanding of what COVID was going to be. So here I'm presenting you with the first wave in India, in Brazil, and in South Africa. In fact, in India, Prime Minister Modi had announced already India had conquered this virus. And you can see that because you see how small their first wave was. Well, the variants changed that. In India, the Delta variant caused a massive wave of infections. I don't even remember. Do you recall watching it on television? People were dying in the car parks because they can't get into the hospitals. The hospitals are packed. The deaths were occurring in their thousands. As it turns out, over 1 million people died. Brazil had a new variant, the Gamma variant, and that led to their horrible second wave. South Africa, we wanted to have our Balito rage for the matrics in the midst of a new variant called the Beta variant. From one Balito rage, over 1,000 cases. And you know, they took the virus everywhere in the country. I didn't know this, but people in Gauteng like Balito. They can't have their own rages in Joburg. They come to Balito for the rage. It spread like crazy. And we dealt with a horrible wave of Beta variant and subsequently Delta variant. So what we were learning is that the mutations in the virus were changing the virus and changing the behavior of the virus. And we were among the leading proponents of understanding this because we were the first to describe it. And so we published this paper in the NEJM, one of the first descriptions about how the variants and their genetic changes were changing the clinical impression of what we were seeing, had a public health impact, and had vaccine implications because they could escape. The new variants could escape vaccine immunity. And what that meant is that we no longer had an end game. So the issue was pretty straightforward. New virus, we now have a vaccine, we vaccinate everybody, we get herd immunity, end of story. That's our end game. When we had a new variant, that end game went out the window because now we were dealing with a new form of the virus that escapes vaccine immunity. And so we would no longer have that benefit of past infection protecting against reinfection, like you have with measles and other diseases. And we needed to understand this. And here I was busy trying to grapple with this with colleagues across the world. In fact, I got a call from Hyder Hackman to say, we need to put a team together to understand how we're going to end this, what's the future scenarios. And that's that paper you see with Peter Piot and Peter Gluckman and others, where we were trying to grapple with what are those endgame scenarios. If you don't know what your endgame scenarios are, you don't know what to do now because you need to make your decisions now to choose the scenario you want. Because if you don't know what scenario you're heading for, what scenario you want to have, you don't know how to make today's decision in your response. And so that's where we were beginning to grapple with the scenarios of what it meant to end this. And so in August of 2021, I was being interviewed. I do a lot of interviews. And I was being interviewed by Bloomberg. And this journalist, Pranisha Naidu, very persistent. She asked me, when is the next wave going to occur? I said, you sure we're going to have a next wave? She says, yeah. I said, are we going to have a next wave? And when is it going to occur? And I said, well, you can't predict those things. We don't know when it's going to occur. She asked me, and she asked me, and she asked me. She wore me down. I was convinced she was planning something, like a wedding or something. She needed to know when that next wave was coming. She wore me down. And I said something that I would not normally say. I said to her, based on a very simple analysis, we can expect the next wave on the 2nd of December, 2021. And that wave we expect would last for about 75 days. As soon as I said it, I regretted it. I said, now I'm in trouble. But what happened? exactly on the 2nd of December 2021, South Africa passed the threshold of a wave due to Omicron, and Minister of Health went on television to announce to the world that we are now in an Omicron wave. The next day, social media is abuzz. Professor Karim is spreading the virus. How could he have known it's coming on the 2nd of December unless he's the one that's spreading it? So much so that Newsweek went and did a fact check on me. Did I really say this? Don't make predictions. That's not a good idea. Even if they come right, they have huge consequences. But because Omicron came, and we, at that stage in South Africa, were really thinking way ahead on the variants because of the number of studies we'd done, I was on television being interviewed about this new variant, just because we just announced it. And I get an email from the editor of The Lancet. He says, "Slim, you're everywhere on television. You're on Sky News, you're on BBC News, CNN, talking about this new variant. You've got to write something." I said, "But we know so little about it. We just discovered it. What can I write about it?" He says, the scientific community needs to know. So Croatia and I sat down, we said, okay, what can we share that will be useful to this world scientific and medical community? And so we wrote this paper. We wrote it in less than 8 hours. We had our 2 statisticians working on the diagram, and we wrote it because they wanted it straight away. We sent it off to the journal. Within the hour, we got back the reviews and the edits. They gave us 45 minutes to address it. We fixed it. We sent it off, the final version with all the corrections, at 5:45 in the evening. Half an hour later, it was online. This is now my second most cited paper, well over 1,000 citations, because it was one of the first papers to be published on Omicron variant. And what it provided the world with the most important information they needed, which is its doubling time. Can you see how that red line, the Omicron line, is straight up? That virus is spreading like crazy. All of the other waves, the Delta wave and so on, they look pedestrian compared to this new Speedy Gonzales in Omicron. So that's what everybody needs to know. We are dealing with a really fast-spreading virus. A few weeks later, we produced these data, which we shared again with The Lancet, showing that Omicron is not as severe. You get a lot of cases, but the number that's admitted is much smaller. Can you see? It's only at 5% compared to Delta and Beta. The other 2 bars are Delta and Beta. And even if you get admitted, the proportion that need oxygen is much lower, and the proportion that dies is much lower. So we are providing the first set of evidence to suggest that this is a much less severe infection. It spreads fast, less severe. And then we provided, this was the first study that looked at whether the Omicron can escape antibodies, vaccine antibodies. This is published in Nature. And here we showed how those who'd been vaccinated with the Pfizer vaccine, Omicron cared little for those antibodies. It would simply just break through. And so our vaccines were not going to work all that well in protecting us against Omicron. And as we know, that's what happened. So I've given you a sense of 30-odd years of research on HIV 3 years of research on COVID, let me just leave you with one lesson on my last slide. A key lesson from HIV and COVID-19, and that is the importance of our mutual interdependence and how we have to have a shared responsibility in dealing with a pandemic. And I'd like to quote here from UNAIDS document. The document is called How AIDS Changed Everything. And I quote, "The AIDS movement demonstrates that with a shared vision, shared responsibility, and through global solidarity, we can change the course of history." And we have. HIV and AIDS was a uniform death sentence. Today, we can get antiretroviral treatment to the most remote village in Africa. We have changed that course of history. Why? Because we cared for our fellow human beings. We didn't say to ourselves, "Oh, no, I want to go to the beach, and I don't care about what everybody else, so I want to do this and I want to do that." We have to think about everyone else as well. My risk of infection affects your risk of infection. Your risk of infection affects my risk of infection. By the way, if I had COVID, I've probably infected all of you. So we have to find our humanity. We have to find the point where we care as much for others as we care for ourselves. Because if we don't, we're not going to survive and do much better in the next pandemic. It's a shared responsibility. We all share it because our risk is so interdependent. And that's so well captured in the phrase that's so widely used in COVID, that no one is safe until everyone is safe. Thank you.
Thank you, Prof. Karim. I know we're running out of time, but we'll take one or two questions from our audience here. So let's give, yes, the two ladies. Yes, please go ahead, introduce yourself. There's a mic coming, hold on. Yeah.
Thank you so much for an awesome lecture. It was really great. I just want to know, how did you find CAP 256? Between everyone all over everywhere, I just think that is the most awesome question I would like to get an answer to.
Shall I answer it? Actually, I wish there was a profound answer for you.
Serendipity.
But we had not known about broadly neutralizing antibodies. It hadn't been discovered. In 2010, two groups, one at the Vaccine Research Center at the NIH and another at Scripps Institute in San Diego, both published papers in the same issue of Science describing broadly neutralizing antibodies. So suddenly we learned, my goodness, there's a broadly neutralizing antibody. We didn't even know it existed until then. Luckily, we had a student already there, and that student spent a month in the lab learning the technique of how to find broadly neutralizing antibodies. So when she came back, we sent her thousands, not thousands, a few thousand of samples. And we said, screen them to look for a broadly neutralizing antibody. In screening all those thousands, we found 8 people with broadly neutralizing antibodies. 7 of them weren't particularly good. CAP 256 is a different story altogether. Very unusual. And we don't fully understand why she's so unusual. But what we do know is that the lineage of the B cell that makes CAP 256 antibodies. So you remember that, well, let me not say remember because some of you might not remember, but the way antibodies work is that they have these 2 arms that go and attach to the virus. Now in HIV, the 2 arms aren't able to kill HIV generally. because HIV is like a Smartie. You know the sweet Smarties or M&Ms. HIV has a shield around it of sugar. There's a whole lot of glycans that cover the virus. So what happens is that that sugar, like the Smartie, you can't get to the chocolate because the sugar blocks you all the time. And it's the same with HIV. You can't get to the protein surface of the virus because of the sugar coating on the outside. But CAP256 has something very unusual. She has a long CDRH3 region on her antibody. That CDRH3 region gives her extra long arms. And those arms, literally, they go through the holes in the sugar, and they go straight onto the virus, and they attach directly onto the virus and kill the virus. That's what makes CAP256 the most potent of all broadly neutralizing antibodies that exist, that are in clinical development. So you get the sense of how we found it.
Thanks. Let's go to the next question.
By the way, all of this is being done in South Africa, just so that you understand. And with huge partnerships. I've already shown you the partnerships. Harvard University doing our monkey studies, NIH doing product development. I mean, you can see multiple players involved. But all the time, the African researchers are driving the agenda. They are leading the team that's doing this work. And of course, you know, we own the patent.
Thank you, Professor Karim. You've given us an excellent overview of the scientific response to HIV. I'd like to press you somewhat on the political responses to HIV, and then when it comes to COVID, to ask you specifically about why the Minister of Health has continuously refused to make public the MAC reports.
What reports?
The MAC reports, the Ministerial Advisory Committee reports.
Oh, the Ministerial Advisory Committee, okay. Yeah. So just the Ministerial Advisory Committee, the MAC, you want to know the advisories. Yeah, okay, I can tell you about that. That's actually, it's quite an easy question to answer. Traditionally, now you must know, I've been providing advice to many governments, to the WHO, to many, many different players for many years. Science advice is a backroom activity. In all the years pre-COVID that I've been giving advice, I've never gone on to any television to say what I've been giving advice on. My job is to sit in the back room, analyze the data, produce the graphs, do a presentation to the policymakers. They listen to many other people as well, and they make the decisions. The decisions are not owned by me. My advice is owned by me. I'm not responsible for the decisions. That's their decisions. COVID changed that. So what is traditionally a backroom activity, in COVID became a front room activity. Not just a front room activity with bright lights on it. Right? And it's a real problem, because if you bring 50 scientists together, you know you're going to have 50 different opinions. It's in the nature of science. It's what is the strength of science, is that we differ with one another. And we differ a lot with one another. Now, I remember, you know, when I joined University of Natal as a Deputy Vice Chancellor, I went to my first Senate meeting, and I was blown away. Nobody can agree on anything in University Senate. I mean, if you bring all the professors of the university, which is what the Senate was at that time, they can't agree on anything. And it's true in the MAC as well. These are strong-minded people. They got strong ideas. we differ with each other on so many things. At the end, we come together and we find a path and we move forward. And we used a very simple approach. We took sufficient consensus, and we had to provide an advisory, and we provided in the first year 114 advisories that we all agreed on. But we disagree with each other. When the public sees that, what do they think? The scientists are fighting. That's what they think. They don't understand the nature of academic discourse. That we'll argue to death, and then we're going to have a drink together. Because in their family squabbles or in politics, when they fight and debate with each other, they're going to puncture somebody's tire afterwards. It's a different model. I was very hesitant to make the MAC advisories available. I was very hesitant because I didn't know about whether advice will now get confused with policy. Because we will say, this is what we said, but the policy will be different. And we didn't want to create confusion. So eventually I had an idea, which I shared, which is I said, why don't we provide the advisories about a month later? So we make it available, a month late. And that way the government has had a chance to look at the advisories, make all their decisions, and the policy comes out first, and then the science advice that underpinned it can follow. But I could understand the public wanted to know more, and they wanted to know it in real time. And so I had to concede that in the interest of transparency, that's a pretty good idea. And so I changed my view on the subject. But I can tell you that it then became a battle of policymakers versus scientists. And it's a battle that people need to be very careful about. If in all the advice I give, if the policymaker takes even 50% of my advice and converts it into policy, I'm very happy. Because policymakers need to listen to other voices as well. If all policy was just science, why do we need politicians? Why not I just write the policy and make it, that's the policy. Remember, scientists, we are not accountable. We are not accountable to the electorate. We are not voted in. We only follow our professional ethics. It's the decision makers that are elected. They have an accountability. They must make that decision. And they don't always have to follow the science. And in fact, I remember this debate I had with the president. Our advisory said, close the places of worship. And what happened? A delegation of priests went to see him, and they said, you understand that this is causing mental health. People are anxious, people are worried, the deaths are occurring, and they can't come to the church. The president chose not to follow our advice. He chose to go with allowing the churches to be open, but only 50 people maximum. I said, where do you get 50 from? He had to make a compromise. It's in the that's his job, luckily, not my job. My job is just to tell him what the science says. But I would not want the public to now get confused that we said close the places of worship, the policymakers are making a different decision. They have to listen to many voices. And they have to listen to the ordinary people as well, and not only do what science says. So it's a balancing act.
Okay, well, we've got one...
Sorry, I give you long answers.
Yes. Prof. Karim, I have one online question. It says, please comment on vaccine development success for COVID-19 compared to HIV and malaria.
HIV is a damn hard virus to work with. It's so difficult because the enzyme that the virus uses is called reverse transcriptase. Humans don't carry this enzyme. We don't have it. So HIV has to carry the enzyme itself. Reverse transcriptase takes the RNA from HIV, converts it into DNA. And that DNA gets integrated into our host DNA. But when evolution or God, whatever takes your fancy, designed the reverse transcriptase enzyme, it did a really bad job. Because reverse transcriptase makes a lot of mistakes. Because normally, enzymes that read nucleic acids do a very good job. Remember, it's like a zip. It's an exact match. Every time there's a thymidine, there's an adenine. Right? It's a very exact way in which RNA and DNA is duplicated. Except when it comes to reverse transcriptase. That damn virus, that damn enzyme, it makes all new viruses. So none of the children look like the father or the mother. The viruses differ all the time. In fact, in HIV, if I sequence your virus today, if you're infected with HIV, and I sequence it a month later, the virus will be drastically different. And you know this, how? HIV is one of the few diseases that you do an antibody test for the virus. Normally, if the antibody is present, there's no virus. Right? The antibody kills the virus. But because in HIV, the antibody is killing a virus that doesn't exist. Remember, the antibody is only killing the virus that infected you. So all the new viruses you got, they don't care about this antibody. So we know we have this challenge in HIV, and that's why we can't make a vaccine. We can't make a vaccine because the virus is changing all the time. So now we're trying to use BNAbs, these broadly neutralizing antibodies, as one strategy. SARS-CoV-2 is the exact opposite of HIV. It has a proofreading step. So when it copies, firstly, it doesn't have reverse transcriptase, so it doesn't make DNA. It just copies RNA into RNA. And it has a proofreading step. So it ensures the accuracy and the fidelity of the progeny. So the children are all like the father or mother, whatever you want to call the original virus. They don't change that much. And because they don't change, the virus itself is very stable. And so you can make an antibody response against it until you get a variant, of course. And the variants are very interesting. I can tell you a whole story about how variants are created. But because the virus is stable, the antibodies against the RBD, that's one section of the spike protein, when that part of the virus is attached by an antibody, it kills the virus. And we know this quite well. And why is it you can make a vaccine against SARS-CoV-2 in 11 months? And we've been working on HIV vaccines for 40 years, and we don't have one. Well, it's because we didn't make the SARS-CoV-2 vaccine in 11 months. That's a common misnomer. I read Katie Carrico's papers on mRNA, more than 20-odd years ago. She had been working on mRNA all these years. You know, she's going to get the Nobel Prize, right? It's with Drew Weissman. She's winning all the prizes right now because of her discovery on mRNA vaccines. She was struggling so much with her work. She's a Hungarian biochemist. That actually she couldn't get tenure, and eventually the university she was at fired her because she couldn't get grants, because nobody wanted to fund mRNA research. And eventually Drew Weissman at Penn attracted and told her to come and work with me. I'm working on mRNA vaccines. And so they have been working on mRNA vaccines for over 2 decades. In fact, Katie herself has been working on mRNA vaccines for over 30 years. Barney Graham, a good friend of mine, has been working on coronaviruses for over 3 decades. He's at the Vaccine Research Center of the NIH. When COVID first hit, he was the first call I made because I knew he's the expert. Well, he made a vaccine against the original SARS. You know, the first SARS. That was in 2002. He had a vaccine, he made it. But by the time he made it, there was no spread of the virus. The virus had gone. And so it was sitting on his shelf. So he took that sequence, he adjusted it for the furin cleavage site, and that's the sequence that's in the vaccine. That's what's in the Pfizer vaccine, that's what's in the Moderna vaccine. So all of this work, when people tell you the vaccine was made in 11 months, it was made from work done 2 decades earlier. Because you can't make a vaccine in 11 months. It can't be done. You have to have the platform technology. Now that we know how to make mRNA vaccines, when the next pandemic comes, and we can anticipate it's going to be sooner rather than later, we can probably make an mRNA vaccine within 3 to 4 months. That's how quick we can do it. Because the platform is there, you just got to insert the right sequence. That's all. Provided you know what the immune correlate of protection is, you can get it very quickly. So that's how we made SARS-CoV-2 vaccines in 11 months. By not telling you about the other 2 decades that preceded it.
Thank you very much. And I think, colleagues, I think we can go on all night talking on this interesting and exciting topic. But I'll ask Prof. Arieti to close off this evening's function. Thank you, Prof.
Pleasure.
Distinguished ladies and gentlemen, I mean, from the applause, it's obvious to me that you are all quite satisfied with the choice that we made. So thank you very, very much. What I didn't tell you in the introduction was when I met Salim in Cape Town and invited him, and I gave him the reason that his work was globally acclaimed, and therefore he would be the right person for us. He said to me, you know, maybe you should invite my wife instead. He said, maybe you should invite my wife instead. She does all the work. And today, I noticed from a lot of the publications that he showed us that she was always present where he was present. It's a wonderful team, and one day I will invite your wife also. But I think we've had a wonderful evening. It's not often that you listen to a researcher, a scientist, that you understand almost everything that he said. I'm an economist, but I've understood everything. And it shows you the caliber of scientists that we're working with, the type of scientists that we want to see more of in Africa, the scientist that's able to speak across generations, speak across disciplines. And we've seen that today. So it's not surprising that the work that he and his team have been doing has attracted the kind of attention that we've seen. So we thank you very much. We thank you for doing us the honors. We thank you for indulging us. We thank you for making time. We know you're a very, very busy man. You were humble and very modest in accepting and making your way here. We appreciate what you are doing for Africa and what you represent for African scientists. I do hope that all the young African scientists watching you online have learned a lot, not only from the science, but also from the presentation, how to do a presentation across disciplines, and for people to appreciate that. I'm quite sure even policymakers like what you say, because they do know that ignoring what you say can be extremely costly. So we thank you for all of that. We couldn't have asked for a better. It's not often that I go to events and then I see people being asked to applaud the speaker, and I don't like it when I'm told to clap for the speaker. But today, I have no difficulty at all asking you to clap for the speaker again. We are extremely happy with what you've done, and we like to show our appreciation through a few things that. This is from University of Pretoria. So I pretend to be the Vice Chancellor of the University of Pretoria.
Thank you.
And this is from ARUA. I carried it all the way from Ghana.
In Ghana.
So with that, we come to the end of the first ARUA Distinguished Annual Public Lecture, delivered by our own Salim S. Abdool Karim. So we thank you, and do look forward to seeing you again at the next lecture, which will be sometime next year. We will announce that. Thank you.